Hyperglycemia in pregnancy is known to increase the risk of complications in both the mother and the baby. For example, women who experience gestational diabetes have a 26% higher chance of developing heart disease in the future. However, the underlying mechanisms by which these defects occur are still unclear, including the uncertainty of whether these complications could be linked to irregularities in the mother and child's DNA. If we had evidence that hyperglycemia in pregnancy does damage the DNA, then it would suggest that the DNA could be a target of focus in the prevention and treatment of diabetes-induced diseases. A study was conducted on 144 pregnant women to address this knowledge gap. It was found that there was roughly a 4-fold increase in the nuclear DNA damage in the maternal cells when looking at the group characterized by hyperglycemia. In contrast, there was less than a 1.5% difference in the DNA damage levels between the control group and the hyperglycemic groups of the fetal cells. Therefore, these findings suggest that hyperglycemia likely produces damage on the DNA of maternal cells, but fetal cell DNA is protected from the negative effects of hyperglycemia in the intrauterine environment. This information implies that the DNA may not be the source of health complications associated with hyperglycemia in the fetus, which allows researchers to have a better understanding of the mechanisms underlying the defects associated with hyperglycemia, because it narrows down what they should focus on in preventing and treating these complications.